KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Neet, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing Megasporogenesis. Megasporogenesis, and one more is development of embryo sac, or female gametophyte information also we will see. So the maximum information we know very well, but a few points to be learned. Okay, right. Now first of all, Megasporogenesis, the formation of megaspore or the development of megaspore. How this particular process taking place, now let us see. Now the process of formation of megaspores from megaspore mother cell is called Megasporogenesis. Means once you say that the process of formation of megaspores. So means here the development of megaspores we are observing, and these megaspores are developing from the megaspore mother cells. Okay, that particular process we will be taking as Megasporogenesis or Megasporogenesis. Now during this development or during the development of ovule, in the beginning of this process, new cells develop from placenta. So first of all, new cells development, how this particular new cells development taking place means, new cells develops from the placenta in the form of small rounded outgrowth like structure. So means new cells development. So this information we don't know, we have to learn this information. During this development of ovule, so what is happening? What is happening means, in the beginning of this process, new cells develops. Which one developing or which one's development is there initially? New cells. So new cells develops from the placenta in the form of small rounded outgrowth like structure. Means here how it will be, small rounded outgrowth like structure formation taking place. That one only we will be taking as new cells. At this stage, all the cells of new cells are undifferentiated. Means in this particular case, no, what are the cells are there in the new cells? No, all these cells are undifferentiated. Means we could not find out any kind of differences between the new cells. So thereby here what is the point? All cells are seems to be same. Just now new cells are formed, and all these cells are how it will be, the small rounded outgrowth structures, small outgrowth like structures, and here the cells all are undifferentiated. So when undifferentiated meaning what? So here the all cells are seems to be, or the all cells are morphologically similar, seems to be same, and you see there homogeneous and meristematic. So here when these cells are homogeneous, so homogeneous means when the cells are seems to be same and having meristematic nature also, my dear students. So meristematic nature means definitely these cells has the capability to divide. Meristematic cells or division of labor or the division of capacity is there only for meristematic cells. Okay, so first of all, once you see there, First of all, nucellus development taking place from the placental cells, and when this nucellus development is there, no, that particular cells, how it will be nucellus cells, initially small rounded outgrowth like structures are there, and all these cells or nucellus cells are undifferentiated, means seems to be same, is nothing but homogeneous, and has the capability to divide, means meristematic nature, and finally they become parenchymatous. So meristematic tissues are there, no, and so finally after a few divisions, no, definitely, so which one formation taking place? Parenchymatous cells formation taking place. So this mass of cells, which one group of undifferentiated cells or the homogeneous cells are there, no, or the parenchymatous cells finally are there, no. So this mass of cells is surrounded by single layer epidermis. So means here all these group of cells are there, all these group of cells are commonly surrounded by only single layered protective layer, is nothing but epidermis. First of all, placenta is there. From this placenta, which one formation? Nucellus cells formation, and these nucellus cells, no, small rounded outgrowth like structures are there. Undifferentiated cells means all cells are seems to be same and having meristematic nature. Finally develops into parenchymatous. Later all these mass of cells are surrounded by single layer epidermis. Single layered epidermis. Next one, any one hypodermal cell of nucellus differentiates and increase in its size, means here the group of cells are there, or the mass of cells are there. Mass of cells means so many are there. So there out of so many number of cells, no, one hypodermal cell means, why here the other mentioned as a hypodermal cell means, what are the cells which are situated just below the epidermis, we will be taking them as hypodermal cells. So means definitely this particular nucellus also is present below the epidermis only, no, that's why here we have written like that. So means one hypodermal cell of nucleus differentiates and increase in its size, differentiate and increase in its size, and it become different from the rest of the cells due to the presence of a distinct nucleus. Means first of all, a parenchymatous group of cells are there. What are the cells or which are present just below the nucellus, no, one particular cell only differentiates in itself, so differentiate and increase in its size. Differentiation taking place. Once the differentiation is completed, it becomes different from the rest of the cells. Means here definitely it will show the somewhat significance when compared to the remaining cells. So rest of the cells, due to the presence of a distinct nucleus, means if you see there, so what are the main important points, no. So in this particular nucellus cells, we can find out the distinct nucleus, means here the presence, here the nucleus appearance also will be very clear. Okay, that particular cell we will be taking as archisporial cell. Archisporial cell. Did you hear this archisporial cell before? So yes, we have got explanation on this one. We got already explanation on this one. Archisporial cell we seen in the development of anther ones, isn't it? So likewise in Megasporogenesis also, first of all, from the placentation or from the placental cells a nucellus formation takes place, and this from these nucellus cells one archisporial cell formation taking place. So next one, archisporial cell divides mitotically. So this particular point onwards important. So what are the archisporial cells are there, no, all these archisporial cells are divided mitotically to form sera. So one more time, archisporial cells are there, no, these archisporial cells, archisporium cells or archisporial cells divides mitotically to form a primary parietal cell and primary sporogenous cell. So here this kind of information, already same information or the same terms we have seen in the development of anther also. So primary parietal cells and primary sporogenous cells development formation taking place. So now the primary sporogenous cells, appadana second one, The primary sporogenous cells directly acts as megaspore mother cell. So in this particular case, no, what are the primary sporogenous tissues are there, or what are the primary sporogenous cells are there? All these cells directly acts as megaspore mother cells. Megaspore mother cells at micropylar region. At micropylar region, and it divides meiotically. Which one? Megaspore mother cell. This particular megaspore mother cell divides meiotically, means reductional division or meiosis process. So by undergo the process of meiosis, so definitely which one formation takes place? 4 haploid megaspores formation takes place. 4 megaspores or 4 haploid megaspores formation, so we can observe. Means here 4 haploid megaspores are formed from the one megaspore mother cell by the process of meiotic division. Next one, the 4 haploid megaspores are generally arranged in a linear tetrad. So once you see there, so here 4 pollen grains, like if you see there 4 pollen grains in a male reproductive system, so that particular the presence of 4 pollen grains together we are calling as like a pollen tetrad. But in this particular case, no, 4 haploids or 4 haploid megaspores are formed. These 4 haploid megaspores are generally arranged in linear tetrad. Linear means one line madri. Linear tetrad arrangement we will be observing. So next one, generally the lowermost or chalazal megaspore remains functional. Means 4 haploid megaspores are there and the arrangement also is linear. So in this particular case, no, out of 4 cells, what is a cell which is situated or which is present towards the lowermost or chalazal megaspore? Means the particular megaspore is situated towards the chalazal region. Chalazal region remains functional. Means out of 4 megaspores, means here out of 4 megaspores, so what is a cell will be functional? What is a cell which is situated towards the chalazal region? That will be functional, and other 3 cells of which are lie towards the micropylar region degenerates. So this particular information means 4 cells or 4 microspores are there. Out of 4 microspores means 1 minute. 4 microspores are there. All these microspores arrangement is a linear type, and these cells are situated between the micropylar region and chalazal region. Now 3 cells, what are the cells which are nearer to the micropylar region degenerates, and what is the remaining cell which is located towards the chalazal region will be remains functional. So now the functional megaspore produces this particular functional megaspore involves in the development of female gametophyte, or the functional megaspore produces female gametophyte. Means here, what is the point here? One and only the functional cell is remaining, and this particular functional megaspore only involves in the development of a female gametophyte. So thereby, for the development of a female gametophyte, how many number of megaspores involvement is there? Only 1. So thereby, here this kind of development of a female gametophyte development we will be calling as a monosporic type. The development of embryo sac. Why? Because a female gametophyte we will be calling by one more name, female embryo sac. No. So thereby, so here the complete female gametophyte or embryo sac development taking place from the one single functional megaspore. That's why the development of the embryo sac or the female gametophyte we will be that particular type we will be calling as monosporic type. Monosporic type. Next one, in most of the angiosperms, chalazal megaspore remains functional. So this particular point we have seen. In this side, what is information we seen? Side heading is Megasporogenesis. Side heading is Megasporogenesis. Now here in Megasporogenesis, just we are studying the development of megaspores. So first of all, in development, during this development in a ovule, so what is happening? That particular phenomenon or information only we are observing. Placenta is there, no? Where the ovules are attached to the placenta by means of a stalk. That particular placenta is there, no? From that particular placenta, new cell develops first. That particular from the nucellus cells to archisporial cells formation taking place. Archisporial cell now divides and form primary parietal cells and primary sporogenous cells. Now, primary sporogenous tissue directly acts as primary sporogenous cells directly acts as megaspore mother cell. So already I told you in previous classes what is the, suppose if we are studying about the mother means definitely ploidy will be 2n. So means it has the capability to undergo the process of meiotic division. Definitely here chance is there for the process of meiotic division. So thereby those cells undergo meiotic division or the reductional division. So thereby 4 haploid megaspores formation taking place. So these 4 haploid megaspores arrangement also linear type, and these linear type of megaspores so out of 4 megaspores 3 will be disintegrated, and these 3 means these 3 cells also which are nearer to the micropolar region, and one and only the cell will be functional, that is located towards the chalazal region. That particular one and only the cell involves in the development of female gametophyte or embryo sac. A point you understood now. So means in this particular case few terms we need to remember. If you see there in a pollen tetrad we have seen in a male reproductive system, in a pollen tetrad, so like here there is no disintegration in pollen tetrad, isn't it? So that's why here what is the main important point in Neet examinations we have to remember, after completion of meiotic division here in case of a female, so after completion of a meiotic division, out of 4 tetrads, out of 4 haploid or out of 4 linear tetrads, 3 are degenerated. Means after completion of a meiotic division 4 are formed, but one and only the living one. So in case of male one, after meiotic division from the microspore mother cell or pollen mother cell 4 are produced, 4 are active. So those 4 cells only we are calling as microspores, or simply we are calling as pollen grains. Did you remember? So thereby here what is the point, in male reproductive system from the microspore mother cell after meiotic division 4 tetrads or 4 pollen grains of formation we are observing, but in case of a female 4 are produced. I am not objecting that one, 4 are produced, but out of 4, 3 are degenerated, one and only the functional. So that particular one involves in the development of embryo sac. A point you understood here. Now once you see that this particular till now what is the information I have given explanation, small rough diagrams also I have drawn for our explanation point of view. Once you see there archisporial cell development. So here again I didn't draw new cellus and new cellus from the new cellus archisporial cell development likewise. So I didn't draw directly, I have taken archisporial cell from the new cellus cell. Why? Because this is one and only the cell differentiates, and it has which one distinct nucleus. Already we have seen now. So their nucleus appearance also very clearly we can see. So later this particular archisporial cell divides into 2 cells. One is so like a linear arrangement, one above the other is there now. So there one is a primary parietal cell and one more is primary sporogenous cell. Next one, if parietal cells upside, so here next layer so 2 cells are there up and 2 cells are there in a linear there 2 cells are parietal cells, and middle cell we will be taking as so middle cells will be taking as megaspore mother cell. Why? Because our knowledge point of view now only we learn now primary sporogenous cells directly acts as megaspore mother cells we seen. So now this megaspore mother cells, now you see there exactly at spotlight, this particular region we can be taken as a micropolar region. So means this entire process is taking place towards the micropolar region only. Don't forget that particular point also. Don't forget that point. Next one, so this particular megaspore mother cell now undergo the meiotic division. So here, we can write the meiosis process or the reductional process. So thereby linear tetrad megaspores formation we can observe. Means here the linear tetrad formation only taking place when this particular megaspore mother cell undergo meiotic division. Now after 4 cells, you see there is the micropolar region. Now first 3 cells which are nearer to the micropolar region disintegrated. You see there. So only 3 cells are there, but I strike off it. And here the key one cell is down. That particular one and only the cell will be in functional. That particular cell only, our subject point of view, we are calling as a functional megaspore. That is a small information related to the megasporogenesis. If megaspore is there, megasporogenesis means the development of a megaspore only. No, now we are observing development of a megaspore. Now how many functional megaspores are there? One and only the thing. Actually முன்னாடி 4 இருக்கு, 4 are there, but 3 are degenerated now. So now the functional one only one. Functional megaspore, one and only the thing is there. Up to here we can be taken under megasporogenesis. Now the development of megaspores, or the formation or the process of formation of megaspore. Now we see. Now let us start to discuss which one, development of embryo sac or the development of female gametophyte. So here also somewhat important points are there. Listen carefully. Now here, now side heading is megagametogenesis. Which one now here we are going to study? The development of, or the process of the development of, or the formation of megagametes. Megagametogenesis. Sir, megagametes means what, sir? Means already I told you, mega means which indicates a female, micro means which indicates male. Already we don't know. Now here megagametogenesis, mega means you try to keep beside a female. Gametogenesis means which one are going to be developed or formed? Gametes. Which ones? Female. That's why megagametogenesis. Now here you see that it is easy, the explanation, everything easy only once you see. So towards the micropyle region no cells, and towards the chalazial region one functional megaspore. It is cell nucleus. Nucleus, the not cell, nucleus. Now here I have written what is that? No, free nuclear divisions. So here I didn't write even a free also, but through my voice you can get. So here the free nuclear division. Sir, free nuclear division means enna sir meaning ana? So our knowledge point of view, suppose if you take a cell division, there karyokinesis and cytokinesis, likewise 2 types are there. No, karyokinesis is nothing but division of nucleus, and definitely this particular karyokinesis process followed by cytokinesis. But listen carefully, but free nuclear divisions means only we will observe the karyokinesis process, but this particular karyokinesis process is not followed by cytokinesis. Point you understood? So thereby division of nucleus we will study, but after completion of division of nucleus, means the formation of cell wall process is not takes place. That particular process we will be taking as free nuclear divisions. So here the one functional megaspore is there, undergo free nuclear divisions. My dear students, don't forget here what is the process taking place? Free nuclear divisions. So free nuclear. I will revise one important point. In endosperm, in endosperm formation also, one time we have used the term free nuclear divisions. Did you remember this one? So while explanation of endosperm information, so I have used the term of free nuclear divisions there also. So karyokinesis process is not followed by the cytokinesis process. So likewise means here free nuclear division in our subject point of view we are using by second time. Okay, now let me continue. Now here the megaspore, functional megaspore is there. So free nuclear division 1. So here 2 nucleuses are there, not cells, only nucleus. 2 nucleuses are there. When this particular functional megaspore divides, 2 nucleuses are there. These 2 nucleuses are moving moving towards the opposite poles. In a micropyle region and here the chalazial region, 2 polar region. Next one nuclear division 2. Second nuclear division. Now, here 2 cells and here from these 2 cells. Next one nuclear division 3. Nuclear division 3, totally here 4 cells and here also 4 cells. Next one finally, so here male 4 cells are there and here also 4 cells are there. No? From these 4 cells, 3 cells together. Listen carefully. Out of 4 cells, 3 cells together involves in the development of egg apparatus. Egg apparatus is nothing but one egg cell and two synergies. Together we will be calling as egg apparatus. Don't forget, embryo sac means this entire structure. Egg apparatus means this mattam. Amma point you understood? Embryo sac means this LMA, structure LMA, but egg apparatus means this mattam. Egg apparatus means egg cell with two synergids. Okay, so now here what is happening now? So now here one important neat bit let us discuss together once you see. It is a fact or it is of interest, to note that these mitotic divisions are strictly free nuclear divisions. That is, nuclear divisions are not followed immediately by the cell wall formation. Means here, so nuclear divisions first taking place. After that one, once you see here functional megaspore is there. From here to two nucleases are there. Here there is no chance to the cell wall formation. When entire nuclear division is completed, then only we are observing the cell wall formation here. But here two nucleases are there. Here there is no chance for the formation of cell wall. Here also no chance. Here also no chance. Once the cell division or free nuclear division is completed, then only we can see the development of cell wall formation. I hope my dear students you understood this point. Now let me conclude this video. Now the egg apparatus means one egg cell and plus two synergids. Now here egg cell ploidy as well as two synergids ploidy also is haploid. Haploid means just two sets of chromosomes only are present. Next point is polar nucleus or secondary nucleus or a central cell condition is diploid, means two sets of chromosomes are there. Next one formed by two polar nucleuses. Now paringa mele ponga one more time. Here four are there no. Out of four, one cell center go. Here also four cells are there no. One cell center go. So inge one. So both together involves in the development of polar nucleus. Next one, three antipodals which are located or which are situated towards the chelacal region are also haploids. So in this particular case, if you see the structures inside of embryo sac, just polar nucleus only diploid in state or diploid in condition, remaining all cells will be in haploid condition. Next one, cells arrangement. Cells arrangement once you see the diagram. So male 3 are there and the middle how many number of cells are there? Polar nuclei. So male 3 and down 3 and here 1 here two nucleuses are there, just two nucleus but commonly covered by one wall, one cell wall. So thereby once you see there 3 1 3, so thereby cells arrangement is 3 plus 1 plus 3, but nucleus condition or the nucleus arrangement is 3 plus 2 plus 3. This is important, listen carefully. Cells arrangement point of view, 3 plus 1 plus 3. That's why here a total number of cells how many are there? 7 celled stage. And here nucleus arrangement point of view, 3 plus 2 plus 3 means here how many number of nucleuses are there? 8. So thereby in a matured embryo sac, total number of cells are 7 and the total number of nucleuses are 8. Nucleus 8 are there and the cells 7 are there. So that is a main important point. Next one, this type of embryo sac is called as polygonum type. Till now we have seen so totally that particular arrangement of or a type of embryo sac we will be calling as a polygonum type, discovered by Strasburger. This particular development of embryo sac discovered by Strasburger scientist. Strasburger in polygonum plants. In polygonum plant is an example of a plant polygonum. In a polygonum plant, Strasburger scientist discovered this particular development of embryo sac. That's why this entire development of embryo sac we will be taking as polygonum type. Next one, this type of development of embryo sac is called as monosporic type. Why monosporic type here? Monosporic type means here entire embryo sac. If 7-celled 8-nucleate condition, entire embryo sac development taking place from the or takes place from the only single functional megaspore. That's why monosporic. Monosporic means here only one functional megaspore involves in the development of complete embryo sac. That's why this particular development we will be taking as monosporic development. Monosporic development. So this is information regarding which one? Development of female gametophyte. Suppose if a chance is there, so in the next video definitely I will come with the remaining information. So conclusion. So listen carefully, so don't skip anywhere, but so definitely which this one helps for your NEET examinations, so learn carefully. Okay ma, so thank you and be safe at home.